So let's start. It's nice and quiet here. I think the reason is that today's lecture will be, if you've followed neural models of cognitive processes, who of you have? No? Then this is sort of, there's the one lecture that I give in this course that I teach, neural models of cognitive processes. That is this lecture. So if you're going to follow that course, you're going to get something similar to this. Any questions about, reading assignments, practicals, anything like that? Let's get it out of the way. I'm still waiting for the exam date. Otherwise, I'm good to go. I haven't had any coffee. I'm going to participate in some colleagues' research this afternoon, and I'm not allowed to drink any coffee today. I'm the world's major coffee addict, so if I become incoherent at any point, it's likely due to that, not that I'm having a seizure or whatever. So this week has been about, up until now, has been, and I mean in this course, not about world affairs, has been about this idea of trying to get at information processing in the brain more than just blobs of activation, right? I think you understand what I mean when I say that, right? And on Wednesday, what I talked about is to try to analyze, to use a smart machine learning type techniques to analyze patterns of brain activity. And that is a beautiful thing to be able to do, right? And I've sort of given you an idea of where that can go, where that development has taken us. But now I want to give you a different view. That is that of encoding models. So that, and I'm going to explain this in more detail, that if you do this decoding where you have a smart classification algorithm that teases apart these patterns, there's a limit to what it teaches you about how the brain works, even though it can look really cool. So let me get into that, and I'll become clear what I mean by that. So we're going to look at explicit encoding models. But first, let me talk a bit. Oh, still in Zoom apparently, so this thing doesn't work. Yay. So in this decoding thing that we discussed on Friday, right? The main idea is that we're not using the mean signal intensity in a blob of voxels, right? But that we're focusing on this pattern of the activity there. And this pattern is going to be the fodder for this machine learning stuff that we are going to do, right? So again, just to reiterate, you're going to have this training set. So you're going to train this classification algorithm on a training set where, for example, you want to see whether from the pattern of brain activation in some brain region, which I've here depicted as a square sort of randomish pattern, we want to decode whether this brain region does any processing that's related to the difference between stimulus 1 and stimulus 2. Yeah. So if there's any stimulus processing that's different between stimulus 1 and stimulus 2, this classification algorithm should be able to pick it up if I have good data. That's the idea. And so there's two stages, of course, to this type of analysis. That is, that you first train this classification algorithm. It needs to learn what that pattern is for it to be able to distinguish the pattern. And then afterwards, you present that classification algorithm after training with test instances, and you ask the algorithm, which of the two stimuli is this? Yeah. It only gets to make, there's just a choice, right? It's the stimulus 1 or stimulus 2. And then, of course, you can assess the performance of the algorithm, and that is then a proxy for the existence of structure in this brain region as accuracy, and that's just percentage correct, right? And if I have a nice and balanced data set, then chance would be 50%, and anything above 50% would be this classification algorithm having learned some pattern in the data, and that pattern exists is then evidence for the idea that some processing happens in this region that relates to the difference between stimulus 1 and 2. So you see that there's an extra level there. Yeah, we're only in principle, this whole analysis only speaks of a pattern that it's picking up on, and that's really the crux that we'll get into today. So usually people have no qualms with this. That's just the machine learning part of it. But how do you then draw conclusions from having found a pattern to neural mechanisms? And that's where we take an inferential step that some people think is hairy sometimes. Yes. So this doesn't only work for stimuli, right? This can work for mental states, for different conditions in an experiment, right? And again, just to highlight that logic, if there's information regarding this input space, which could be images, sounds, or whatever, then this classifier can successfully decode. So there is a brain pattern, is what this means. Yeah. Now, one thing that has crept up even from the beginning of this this sort of methodology in the field is that if you're leaving the important aspect of this type of research to some smart algorithm, then that may be cool, but it's then also very difficult to understand what is the pattern that it's picking up, right? That can be a sort of a black box thingy. You've left all the hard work, the heavy lifting, to a very sophisticated algorithm, and then if you're just content with this percentage correct measure, then you're sort of giving up on what was that neural mechanism that led to the pattern that this classification algorithm could pick up, right? And that's really the interesting part. So there's a bit of a problem there. Yeah? And on top of that, there have always been, really from the get-go, so from 2004, when these types of things started being applied to fMRI, there's been strong debates as to what type of pattern are we picking up. So when people started doing this, they sort of said, like, okay, we think that fMRI is sampling subvoxel information. So if there's 9 neurons in a voxel that do the color red, and there's 12 that do the color green, then we're going to get this slight offset in activation for red and green. And if you have that sort of imbalance across many voxels, then that's so you're really looking at what the neurons are doing inside each of the voxels, and that's what's leading to the pattern. There's also other people that thought, but we don't really know that this is really the case. It could be that it's just that the back of the head has more blood flowing through it than the front of the head or something like that. And then the patterns actually originate from something that's more similar to that. How do we know that it's really evidence of, picking up a neural mechanism, finding a pattern like this? Yeah? So there's this step that we're taking in terms of our inference. People have been pointing to specific problems in this approach really from the beginning. And it really centers on this question, like, how do these patterns relate to neural mechanism, right? And if we are leaving this question to this pattern finding question to some machine that we don't care what it's doing, that's actually sort of abdicating our what we want to be doing as scientists in some sense, right? At least I tend to agree with this, being critical of this type of thing. You really want to understand each and every step that you're taking, because in the end, you want to understand what the brain is doing. That's, I hope, the goal. Yeah. And that's also why I was talking on Wednesday and saying things like parlor trick, or like that something might be a nice proof of concept, but what do we learn, right? That sort of question is something that I want to really instill in you, is always very important to ask, to be very critical of, okay, this looks cool, but what is it really teaching me? Taking that step back and trying to be critical of what you're being shown. Also in these lectures, by the way. So if you have any critical comments, I'm very open to those. So I want to contend that we should be more interested in the single voxel. I think I switched these around. Yeah. So in the first, this pattern classification instance, it's just the distribution of values, so that this the pattern across voxels that we're interested in, right? But what I'm going to talk about from this point onward is that we really want to try to understand what's happening inside every single voxel. Yeah. So the argument here is that the single voxel is important. It's not just something that you have to average over to get a blob, right? And you shouldn't also just, group the single voxel in a pattern. What we should do is value that this single voxel is an independent measurement and really try to learn more about the brain's underlying mechanisms that then lead to patterns, right? So if I understand what every single voxel is doing, I also understand why a pattern arose, right? Because I know that this voxel was high and this voxel was low in activation. That's a pattern. But I also know what caused these voxels to go up and down in their activation, right? So you're really taking it a level deeper. And the idea is that you try to understand where the patterns come from. Okay? So you can see how the responsibilities are divvied up a bit differently, right? So now all that hard work that was being done by the pattern classification algorithm, all of a sudden we have to do it, which is shitty, right? That's hard. On the other hand, knowing where the patterns come from will allow you to do more things, not just say percentage correct, but be far more specific in your inferences about what the brain is doing. And I'll show some examples of that. So if we go to, many of you might agree, like, hey, yeah, we need to know where that pattern came from. But then, okay, but how do we then understand what each voxel is doing? Yeah, and here we're going to sort of flip the coin from decoding what the brain is doing to trying to understand how the brain is encoding information. That is to say, we want to understand what each voxel is doing. So in fMRI, the unit of measurement is, of course, the voxel, but you can actually do the same type of analysis or inference with EEG electrodes or MEG sensors or intracranial electrodes, whatever, right? It's the unit of measurement that you care about here. And we want to make a model of how that unit encodes information. So what that is called an encoding model. And we want to be explicit and understand how the brain encodes information at the voxel level, right? So because once we understand what makes every neuron and not every neuron in this case, although it could be, but every voxel tick, we actually have a full catalog of what all of these voxels like, right? How they encode information. And then the idea is that if you then see a pattern, it becomes a pretty straightforward inference to then say, okay, but now this pattern of bold responses, I know what each voxel likes, right? So I also mean what it means in terms of a stimulus if one voxel has a high activation and another voxel has a low activation, right? Because I know what those activations mean. Okay? And so in this case, if we have an encoding model for each voxel that we've recorded, what this means is that we can invert this process, if you will. You can start looking at a pattern of brain activation and then say, this was the most likely stimulus that was shown to the subject. And not just, this is more like stimulus A or stimulus B, right? But you can actually really reconstruct stimuli in a sort of statistical sense from patterns of brain activation. And we call that encoding model-based decoding. Yeah? So this really is doing all of the hard lifting, the heavy lifting yourself. Yeah? Any questions about this sort of flipping of the approach, right? So this really has to do with looking at that single voxel, yeah?
It's very prone to errors.
Yes.
If you make a small mistake, or you don't consider some external factor.
Yes, yeah, no, yes. So once you're pointing at a very, very fundamental thing that is very, very important. If all of the hard work needs to be done by us, that means a lot of work, right? And it also means that if we make a mistake in setting up the model to describe what each voxel is doing, then we're going to be blind to that entire part of what the voxel could be doing, also in this decoding. Yeah, so you're right. But I think that's also a general thing, right? And you could make similar mistakes in a purely pattern-based decoding approach. But yes, here, if you're doing this, it becomes apparent, right? It becomes very clear to you, like, hey, but wait, I have to do this? Like when you move out of your parents' house, you notice that you have to start doing the dishes, for example, right? You have to start doing all of the hard work to make things run, right? All of the different ingredients you are now responsible for, yeah? I hope you have a dishwasher, by the way. I would never go back. Okay, so let me just talk a bit about brain organization, and then I'll be able to take this idea of this encoding model and trying to explain the unit of activation a bit further. And so what I'm going to be talking about is how the brain processes information, like how understanding the single unit can actually tell us something about how the brain as a whole is working with information, right? So what's in our minds, yeah? So in this case, it's just unit of measurement, so voxel, yeah. But I keep saying unit because I like this idea, like in the olden days when people were doing single cell electrophysiology, so they would lower an electrode into the brain and they would lower it down and down and down and listen, and then at some point they would hear a cell firing. So they would, the monkey would just be, usually a monkey, could be a cat or whatever, they would lower this electrode down, and then you would start hearing cells firing. You're actually listening to the brain activation, right? And then they could be very sure that they were at a single neuron. They would call this a single unit. And I'll be showing you examples of this. So I think the idea becomes clear. So it's related to the unit of measurement. That is to say that really the sort of atom of your measurement, the highest specificity. Okay? So just a quick overview of brain organization, right? So the idea is in general, and I'm showing you an example of a visual system here, but in general, you start out with very low level sensory information, like blotches of light and dark, right? That's what falls on your retina, and that's what your brain has to work with. And similarly, in audition, for example, what comes in is these patterns of phase and amplitude of sound waves, right? And they're pulled out, they're separated from one another, and then that's the raw sort of information that the brain is working with. And similarly, for somatosensation, I'm just giving more examples, but you already get the point. But in somatosensation, you have different types of pressure sensors that tell your brain where your arms are, where your legs are, and that sense, where you are. And all of that incoming information comes in the raw, and your brain has to distill where you are, where you're holding your hands, right? What is that thing in front of me? Is it a face? Who is it? Right. So what you have is raw information entering through the senses, and as your brain analyzes this information, the information processing becomes more and more complex. That's not a sort of simple thing, how that happens. There's multiple different qualities that change as information processing goes from this raw, in this case, retinal stage, to a high level stage. So you get increased integration between action and perception. Right. So whether so if I want to make an eye movement towards something, planning an action towards something visual, so the two have to interface. Yeah. For example, or when I'm reaching for a cup, you'll have somatosensation and motor actions for the hand, also having to interact with vision, because you're looking at what you're doing. Yeah.
So does it make sense to look at single voxel when you abstract processing?
Yeah. So I get your intuition, because it's very easy if the unit records places of blotches of light, right? That's easy to make a model of. But it becomes very difficult if it has to do with like emotions of people's faces or in people's voices, right? That's a very abstract type of information processing. Abstract. Right, so I agree with you that this task of ours, where we're responsible for every single step of the way, that task becomes harder and harder as we become interested in more and more, more deeper types of information processing that the brain is performing. But I'll show you examples today, I hope, that will give you an idea of how this paradigm of valuing what the single unit is doing may, already be sort of going there. Right, so you get integration over time. The things that happen here are like instantaneous, and there's hardly any memory, whereas higher level regions, they integrate over longer periods of time. You get increased multisensory integration. So the higher you're up, the more important it is that the face that you're looking at is also speaking, so that you can interpret what they're saying, and then you can understand that that might be a very valid building block for trying to understand how someone is feeling, for example, right? You're fusing together the timbre of their voice with these slight things that are happening in their face that you're seeing. Right? So you need to integrate across multiple senses, and at the same time, you have all these specific regions that are very, very specialized. Some like motion, some like facial postures. So they're very, very specific in the types of things that they activate for. And at the same time, you have increased invariance. I'm not going to quiz you on these concepts, but I just want to point out that there's this evolution of processing happening. Invariance stands for that on the retina, whether the cell fires depends really strongly on on the location of the stimulus. Whereas if you go higher and higher, it doesn't depend as much anymore on the exact location of the stimulus. And what that means it becomes invariant to location. And similarly, we have a whole hierarchy of face processing regions in the brain. The lowest processing regions, face processing regions, they really, the activity there depends on whether you're looking at a face from this angle or from this angle. Right? So it's not invariant, it's variant, if you will, to facial direction. Whereas if you go to a higher level of processing, the cells there will keep firing regardless of what the exact direction of the face is, right? It has become invariant to facial direction. Yeah? So we see this sort of invariance happening all over the place. So I see a question, so I'm going to try and answer it. So this is blobology, but more general, applying to patterns based on all previous knowledge of what stimuli trigger certain voxels. Yes. So the idea is that you trigger, so that you learn what triggers a voxel to activate, but you do so per voxel, and you don't try to do this type of analysis across the entire pattern. I hope that explains that answers your question. So this is more or less an aside, right? But I think it's important because it gives you a framework for the different things that I'll be talking about, because we're going to look at these encoding models at these different levels of processing. And we're going to see how our encoding models can also vary in terms of how variant or invariant they are, if you will. Yeah. So I want to start with this classical study. I don't know. Yeah, let me check. I once heard that I can, in a Zoom, say that it should use the projector as a speaker, in which case I'm just going to try. So there's a movie in here. If there's sounds at some point, then it's good. If there aren't, no, they're in here. Wow. So let me change that. I'll just change it to the speaker for my on the laptop. So does this work? I'll just rotate the laptop a bit. Are you hearing anything? Because I am. Okay, this is always a problem, right? Sounds and presentations. I'm gonna kill these for now. Oh, here we are. Sound settings, yeah, why not? Can't really see my. Oh, that shouldn't have happened. Okay, so maybe now.
These are these action potentials that are being inspired by a single cell. Something on screen. Okay, sorry that you're not hearing me. How about now? Can you hear me? Also not. Very far away. And maybe that's because the. How about now? Hopefully better. Oh man, this is hard. Hybrid teaching. Okay, so what you're seeing here is two Nobel Prize winners performing their Nobel Prize winning experiment. So they are recording from a single cell in monkey or cat visual cortex. That is like the first region of the visual cortical system. And what they're doing is they're mapping out of the single cell that they're recording from the receptive field. And what is the receptive field? Well, it's the pattern of light that makes this cell fire best. Yeah. So what they've just done is they've found that there's this region here that if the light is there, it makes the cell fire. And they've also found that there's a part of the image here, if it's dark, that also helps the cell fire. Yeah. Very important here is that they've seen that you get, you heard it spike a bit, right? I'm talking over it. The receptive field, so the pattern of the visual stimulus that is required to make this cell activate, it has an orientation, it has a visual orientation. Only when this light bar is like so, it's oriented like so, does this cell activate. So what this means is also that if you were to turn off the screen and you would listen to the cell while this animal watched a movie, you hear the spikes firing, you know that there is something like this in the movie, right? And this is the basic idea of why this recognizing the value of the unit of measurement is so important, right? So if you know this, not only do you have an idea of what orientation is that, and you can analyze that orientation, you can do all sorts of things with that extra information that you now have. It also means that you can understand the patterns in the brain that are being caused by cells, and you don't just have to rely on the classification algorithm to understand them. Yeah. So it's an old movie. So people can now do this with sophisticated techniques where they can record from thousands of neurons at the same time, and they can just show an animal a movie and then distill for every neuron what exactly the receptive field is. What they're doing here is they're picking another cell, they're finding another cell, and this is a cell of another type. The first cell was simple, and this one is complex. The difference between the two, and this is also not like exam material, of course, otherwise I'd explain the difference, but the difference between, like on the slide, the simple cell has actual patches of dark and light that responds to a static spatial pattern. This complex cell, it doesn't even care whether something is light or dark. It just cares that there is a bar in that orientation. So it still has a location. It still has an orientation preference. But this one only whether there is a stimulus with that orientation in there. It doesn't care whether it's a light bar hitting the light part of its receptive field because it doesn't have a light part of the receptive field. It cares about that bar. Yes. So this cell actually, well, if you have an orientation like this, so if you like vertical orientation, the cell like this will primarily only be able to be responsive to directions left and right, right? So left and right direction. But if you look further, and then watching this movie, you'll see actually a motion direction selected cell at some point. I don't know where it is. This movie. This is where it's slide. But there are these cells that might choose movement direction. There are also cells that like a specific depth, right? Like I talked about stereoscopic depth in the previous lecture. There are cells that like things far away or close. There are cells that like red, blue, and green. Right? So, whenever you see something, right, that richness of experience, it's encoded in this pattern of activation of all these cells. Right? And if you understand what all these cells like, you also understand the computation of the brain is performed as a whole. That's the charm. Here. Yes, to point. So there's this entire field of how this develops, right, in childhood, but also already in the womb. In terms of the visual system, which is my expertise, I know that there are what happens in the womb is that the retina already activates, even though the eyes are still closed. The retina actually activates, and what happens is waves of electricity run over the retina, specific types of patterns. And what that means is that, for example, cells in the brain can already sense this electrical activation of the retina. And for example, know whether a pattern of activation originated from one eye or from another eye. Right? So these waves in the retina, they happen independently from the right eye. And what that means is that before you are born, you already have patches in your visual brain, so far removed from your eye, that are set up to like the left eye's pattern together more, or the right eye's pattern together. So a lot of this is development, definitely, but it's important to realize that it's not just visual experience that drives this development. It actually happens very, very early. So there are like totally hideous experiments in the 1950s where they actually raised cats in environments with totally vertical environments, right, to see whether they could test that they even get vertical horizontal cells, right? They do, but to a far diminished extent. And these cats are, they're probably pretty blind. But if you can only see vertical lines, you can't jump onto the table. So behaviorally also this has this stuff. So what you see here, like this cell, it doesn't fire in the particle. Oh, yeah. I will take out the movie so that you can look at it with sound separately, apart from next to the lecture, okay? My apologies. That's shitty.
I don't have any more sounds, I don't think. So this is what these single units are doing. And of course, this is far removed from all of the things that I already said. Like, we're not measuring neurons, we're not measuring neural activity, we're measuring blood, all of that. But if your voxel is small enough, there's going to be a limited amount of neurons inside that voxel, right? And if the vasculature is precise enough, then you will be able to record from hundreds or a couple of hundred or a couple of thousand neurons at the same time with fMRI. And that's what it turns out is possible. Okay? But I'm going to talk a bit more about that later on. So if we look at these receptive fields, you have a region in the thalamus in the center of the brain, and I'll talk more about these brain regions later on in the course when I'm doing the neuroscience lecture thing. But in this region of the thalamus, it turns out that the receptive field structure is very simple. It's just a center and a surround. So these are quasi-circular receptive fields. So they don't have this orientation that we saw, nor do they have this direction selectivity. Yeah? But only in this first visual region of the cortex, right? So in the neocortex, this is the second relay point, so to say, we start to see that there is an orientation preference to these neurons, right? So their receptive fields become more abstract, right? And more precise. So these cells report orientation. So there are other cells also in V1 that report how fast something is changing. So here, this is X and Y, so you get orientation. Here, this is X and time. So this actually responds best if there is a transition at some rate. And then, like we just already saw in that direction selective cells, there's actually a specific region in the brain called MT for medial temporal, where most of the cells, if not all of them, are direction selective for movement. And that is to say, if you say X here and time here, really means that this cell will mostly fire when something is moving at that speed in that direction. Yeah. Now the cool part, and this also has to do with why we can actually measure these things using fMRI. The cool part is these cells, and I'm using the visual system as an example system here, but this holds for all sensory systems, and the idea of this type of approach actually holds even further than sensation. So the idea is that if you do fMRI, if this is all jumbled up in the brain, if all these cells have nothing to do with one another, with their neighbors, that would be shitty. But the real bonus for us as fMRI people is that if you look at a voxel, the neurons inside that voxel will mostly be doing more or less the same thing. Yeah? And this holds specifically for visual location. Yeah? So that is, where is something falling on your retina? Because the cells in the visual system are organized according to this topography of the retina. We call this retinotopy. And this means that we can actually map this using fMRI in humans in a very, very specific way. So what you're looking at is my brain, yet again, but ethics stuff. What you see here is a map of my visual cortex on the right side of my brain, and I've inflated the cortex here. So that's the neocortex, the foldy bits. And I've projected which visual location every voxel on my visual cortex likes. Yeah? Now, this could be a random patch of salt and pepper shit, right? That is well possible. But because of this grouping of cells together, if they like similar locations in the visual field, we can actually understand what's happening using fMRI. Yeah? And so what you see here is that there is this region in the back of the brain that actually has this entire map of visual space, but it's flipped. So this is what visual space looks like. And that means that red is on the top, here red is on the bottom, green is on the bottom in the visual field, and here it's on the top. So what that means is that things are projected in this way into the brain. Yeah. And why is that? Well, that's really simple. Who of you has a camera phone? So if you any camera does a double inversion of the image, right? The things that are in the outside world on the right top, they will end up in the left bottom on the CCD element of your camera, right? So there is this double inversion just in the fact that you're using a camera. Now your eye is a camera, and so your entire brain just flips everything upside down, left, right. Yeah? It's nothing but a physical thing, right? This is just, this is the way the information comes in, the format of the information, the way that it comes in, and our brains just inherit it. And not just the first visual region in the neocortex does so, but you see this structure reappearing over and over, right? So these regions here are split a bit more. We can go into the detailed topography, but for now, the point really is that your brain really has, we've been identifying more and more of these retinotopic maps in the brain. I think we're at 40 or 45 per hemisphere now. So whenever you see something, you have all these neurons firing, right? Orientation, color, and so forth and so forth. But they seem to always be connected in terms of visual space. Yeah. So if you see something happening on the horizontal meridian there, all of these blue patches, they will activate together. Right? And then, for example, this blue will activate if in that stimulus there's motion. And this here, it will activate if you're planning an eye movement towards it, and so forth, right? But this gives us a common format for a lot of brain activations. Yeah. Point here is, if we can measure the units, we can actually start to look at the architecture of the brain, right? Because a pattern that we will be measuring, it will correspond to this architecture. Yeah. If I want to decode whether I'm being shown something to the left or to the right, this is, you can easily understand the pattern of brain activity that would cause. Great. Now, I'm quickly going to show you an experiment that we use to create these maps. It's very, very similar to the monkey physiology video that you just saw. Yeah? So the idea is that we do exactly what this Hubel and Wiesel did with their bar of light, right? You saw them doing this bar of light thing in the screen. We do exactly the same thing. We just do it on our screen in the scanner, right? We ask people to fixate in the center, and we show this really very busy, very strong stimulus in the passing through the screen. Right? Then, of course, the idea can be what activations did this cause, right? And the nice thing is that if you look in the brain, you can actually see that on these maps that we have in the brain, you see waves of activation traveling over the surface of the brain, right? So what this means is we have very structured visual stimulus, very, very simple movie, and we have very, very structured activations in the brain. And you can actually see waves running through the brain if you show a movie of a wave. Yeah. Sorry? No, we are going to look at single units. I'll get into that in a bit. So indeed, what I'm showing you here is still patterns. Yeah. But this was just to show you that we have these maps, right? And when I show a stimulus like this, you will actually see the stimulus traveling across all of these maps, right? And that creates the waves. And indeed, the wave is a pattern. Yes. So just to give you an idea of what our monkeys feel like, this is the experiment in real time, so to say, because fMRI is slow, right? So what I just showed you was sped up about 20-fold. This makes our experiment really simple and slow and boring. Just imagine that you're in the scanner doing this for 1.5 hours at a time. Of course, yeah, because in an experiment like this, you do this brief run of an experiment and you try to do it as many times as possible, because then you can average them and you get better quality data. So that's the idea, right? So people fixate, and we have this very slowly moving bar moving across the visual field. Boring, eh? Yeah. Yeah. So it's this sort of anthill sort of structure, because that's a very strong visual stimulus. All of these cells, they have receptive fields that look a bit like these elements. And so you're activating a lot of cells with this. Other people use like a checkerboard flickering. This one had all these elements, and the color was in there because we not only instructed people to fixate, but they were actually performing a task on the stimulus. So they were directing their attention to the stimulus, and the question was the bar sort of magenta cyan or blue yellow? So magenta cyan, blue yellow, magenta cyan, blue yellow. So they were pressing buttons to indicate the color of the stimulus at the same time, while not making an eye movement. So we had to train people to not like look at the stimulus all the time. Yeah. You had a question? Yeah, that's hard, right? And you're right. In many cases, the fMRI scanner is a very hostile environment to this sort of measurement. But you notice that I'm talking about this as if it's an important thing, right? And so what we've done is we've invested a lot in being able to track eye movements, even in the 7 Tesla scanner, for example, which is important because, well, in many cases, yeah, what you see in the brain can differ like widely depending on what people did with their eyes. Let me. Oh yeah, this never works. Now we're stuck at this movie. I don't know why that is. So you want to do my experiment? Let's have a break now while we'll be back in 5 to 10 minutes or so. I'll just start whenever everybody's back or when I start feeling anxious. And I'll talk about the type of analysis that we do with this, which is slightly different from the standard GLM analysis and is also very different from the pattern analysis that we've also talked about. Yeah, that would be very cool, Philip. So I'm still recording this, so I'm just going to repeat the question. Does the big bar consist of all the tiny bars because the tiny bars activate simple cells and the big bar thus a complex cell? Yeah, that would be really cool. But that's not really the level of detail that we can actually look at with fMRI. So I'm afraid that you could devise experiments to separate simple cells from complex cells and do fMRI, but it's, yeah, I would be afraid to try. So I don't know how much of the lecture I'll be able to finish, but we'll go and see until someone comes in and breaks this off. What you've seen is these waves of activation traveling across the brain, right? And what I want to do now is zoom into every single voxel, right? Because what we're interested in is not these patterns, but it's the single voxel, right? And so what that means is if you have this wave traveling across the screen and then a wave traveling across the brain, if you're one of those voxels inside that brain, your activation will happen at a very specific point in time, right? Sure. Depending on where you are in the brain and the direction of the wave, you'll either activate early or you'll activate late. And what this means is that we can take this bar type movie thing that happens and these waves in their directions and really try to make a explicit encoding model of what portion of visual space a voxel likes. And this is the way that we do this. Oh, here we are again. So we measure a time course like this in a specific single voxel, right? Because there are these waves traveling, and this is just, sometimes these are a bit closer together, right? That's because here it's going from the top like so, the bar, and here it's going from the left to the right. So you can already infer that if these two are close together, it means that this activation happens late in this bar pass, as we call it, and it happens early in this bar pass. Right? And this means that likely this voxel, it likes a portion of the visual field that's at the left bottom, right? Because these two are very close together. What this allows us to do is we can fit a mathematical model. This is just a Gaussian function. It's not very complicated. It's just a normal distribution in 2 dimensions. And a normal distribution in 2 dimensions has a location. Right? So you can change the location. It's just where the mean of that 2-dimensional Gaussian is. That's this. Right? So this x0 is where that thing is. So it allows us to shift this model of the receptive field that may look a bit like this over space. And then there's another one that another parameter that tallies size. So the larger this sigma parameter is, the larger the portion of visual space that this voxel responds to. Yeah? So we can fit these parameters, and this is our best fitting prediction. And we can quantify this, statistically quantify this. How well do we understand this response? And so this gives us a very explicit representation of what this voxel likes. When this voxel activates, we know exactly where in the visual field something happened. Yeah.
Maybe explain again why you say
Yeah, that's a good point. So let's see. So here we have this bar thing going on, right? And here we have the bar going from the right bottom to the left top, and here it's going from the right to the left, right? These, you have to remember that these bar passes are like 30 seconds, right? And so even in the bold response time frame, this is relatively slow. And you will be able to see whether this voxel has its receptive field. We call this a population receptive field, whether it is here or here, right? Because for this bar, it will activate early here, and then and it would have activated late there. Whereas for that bar, they would have activated simultaneously, right? So these different bar directions, they serve to tease apart what location this voxel likes. So we really we need something like this, right? We need this sort of spanning of visual space at this time frame, also at this time scale, to be able to fit them. So specifically, the example that I just gave you is if something is here. So this is the second and third bar pass in the graph. Here, the bar is coming from the right top and going to the left bottom, right? So that's inverted, like left, right, inverted from this one. And then it's coming from it's going from the left to the right. Yeah. So I'll get to you. If you have a population receptive field here, right? If the bar is coming like so, you'll have a late activation. Yeah. Whereas if after that the bar is going like that and the receptive field is here, you'll have an early activation, right? So a late activation followed by an early activation means that two activations will be very close in time. I'm shifting it, sorry. But that's what you see here. Like this activation is very far removed from this activation, but these two are very close together, right? And so based on this exact time course of the response, we can actually be very precise in what specific location in the visual field this voxel likes. We get like a really, really beautiful description. And you see like the R squared here is also very, very high. And it goes much higher than this even in the low level visual system. So yeah, using a stimulus design like this, a very simple movie, but that does have all locations in there and all directions spanning over time, we can actually use when these activations happen and distill exactly what portions of visual space this voxel likes. Yeah, your question?
Yeah, so this is about location and not orientation.
Yes. Important, only location here. Right? That's also why I was talking only about these retinotopic maps. We're only talking about location. And that's also because for location, we know that neurons that like a similar location are close together in the brain. For orientation, that is actually a very tenuous statement. We don't know how exactly the organization of orientation in the brain, which we know exists at some scale, but it's much finer scale than we measure with voxels, and we don't have a very clear understanding of how that then is measured using fMRI. So that's problematic if we start looking at orientation. Yeah, so you can imagine that if this this receptive field, right, it's very small in the retina, it's very small in this first visual regions. But if you go to face processing regions, for example, the receptive fields are very, very large. And what that means is you have a difference in how large the portion of the visual field a voxel likes. You have this difference across different regions. If this one would be much larger, let's say that its circumference would be something like this, what you would have in the time course, you can guess what you would have in the time course, right? Open question. Yes, you get wider humps of activation. Because the stimulus will just come into the receptive field earlier, right? And then continue and then exit later, right? So you get an earlier activation and a later activation. So if I had taken this voxel, not from V1, where it actually came from, but taking it from high level, like somewhere in parietal or frontal lobe, you would actually see these whoop, 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 and these would it would be a small dip and it would go like so. So there's this beautiful difference there. The size actually means something, right? It has you see that you can actually explain these bold response time courses really well by changing these parameters. Does that clarify things a bit? Sorry, I'm not. So, yeah, I can get it. I can explain. Yeah, so this is a small receptive field, right? So this is a small population receptive field. Yeah, in the brain. Yeah. So this is the spatial pattern in the screen that makes this voxel activate. Yeah. So you, at every time point, you take this this model, right? You multiply it with the stimulus, and then you get a prediction of what would happen in the bold time course. Now, of course, if this is larger, let's say it's the size of the entire screen, then it would just be, is there a stimulus on the screen, right? Then the stimulus is on the screen, like on, off, on, off. It would be very wide activations, right? So that would be the largest possible receptive field. Yeah. Yeah. Yeah, so that's a good point. So what we're interested in is there's this the difficulty in I'm showing it really fast, right? So then you would think like motion direction and orientation matter, but we're only interested in location, right? So that falls out. You can do experiments in which, for example, you want to map out orientation, and you can do. I'll show you that in a bit. But here we're only looking at location. Oh, yes, we can. This 8 is just a historical artifact. People always do 8 because, you know, we want to also appreciate the diagonal directions, but it has no meaning. Like, if you want to be quick about it, you can do half of this experiment and it's still the same. Yeah. No, so good point. Yeah.
Maybe I'm a bit slow, but we said it.
I think I'm a bit slow today, but yeah.
You said when it goes from left to right, you see it earlier, right? So it activates quickly.
No, not all of them, right? So you know that the like, for example, the left hemisphere of the brain likes the right side of visual space and vice versa, right? So that means that you'll predominantly see in the right hemisphere that these voxels like left visual field positions. And so then for a bar moving in that direction, they would activate early. So
this signal at the bottom is one voxel.
Yes, it's only a single voxel.
So we respond better to faster to the bar or right.
Yeah, if the spatial model is that it is here, for example, then yes, yeah.
Then I guess my question is, why is it also so quite a quick response when the bar goes right to left?
Oh, now you're confronting me with something I can't tell you. I don't know.
Okay.
Yes, you would. Yeah. So what's not in this figure is exactly when these bars appeared on the screen. And so, yeah, you're right, definitely. And so in an ideal world, you would take this entire time course, and for every dot in the time course, you would actually show what's on the screen at that moment. And then what you're saying should definitely be the case. From this, I can't really say. Yeah. But that's usually what I do. And then, yeah. So, we now have, right, we can focus on the single voxel time course, and we can relate that to the information processing that is happening by the neurons inside this voxel, right? And we can make this more complicated still. I understand that this already might feel like a complicated model. People make more complicated models, right? So you can do more complicated things. We do more complicated things. It's just to say that you can make very intricate, you can gain very intricate knowledge about the neural processes that happen inside each voxel when you do this sort of thing, right? So there's an enormous power in really trying to model what every voxel is doing. Of course, if you have very crappy data, this sort of thing becomes really hard. And that's also why we're getting better and better scanners, right? So that this sort of thing becomes easier because our data will become better. So I'm not going to talk about specifically this model, but you sort of get the point. Now, I want to take a step back and say, okay, now let's say we have a model of what every voxel in the visual system in this case, right? I'm using that as an example here, but the principles are general. If we have this description, right? We have this whole retinotopic map outlined, and we know for every voxel exactly the portion of the visual field that it likes. What we should be able to do is now take a pattern of bold responses and say, this stimulus was on the screen. Yeah, that's what we should be able to do. And this stimulus was on the screen is a much more precise and rich statement than, 55% correct classification accuracy, right? It really is reconstructing a stimulus from a brain. That's what this encoding model approach allows you to do. And so I just want to highlight how we do this. And I'm going to use Bayes' theorem for this. I'm not going to go through the intricacies of this, but in this case, if we look at, I'm sort of assuming that you've had statistics, right? And I'm not going to quiz you in the exam on Bayes' rule or anything like that. I just want to show you how this helps us structure this idea of being able to decode from an encoding model. Yeah. So S in this case is a stimulus, and B is a bold response pattern. I'm not caring about these two marginal probabilities, the prior probabilities. They're not interesting for this in this case. In many cases they are. But in this case, I want to relate these two things. So this is the probability of a bold response given a stimulus, right? And B is a pattern of bold response, right? So that's activations over a bunch of units, a bunch of voxels. Yeah. Now, this probability of a bold response given a stimulus, well, we've recorded the bold response, right? That's also why this falls out. We've recorded the bold response, and we have shown the stimulus, right? So we have knowledge about both sides of this coin. It's really what our training set gives us, because we know these bold patterns and we know the stimulus, right? So we can now create a model of this probability distribution. Yeah, that is to say, we want to tie the bold response to stimuli. There's a bunch of complicated statistics in there that I'm not going into because it's complicated and it's not part of the course. But what goes into this calculation of the probability of B given S is these bold responses that we have, the stimulus that we have shown. We know exactly what portions of the visual field each of these voxels likes, and we have to do stuff with voxel covariance structure and all of that. Complicated statistics, and we won't go into that. The point is, now we can go in the other direction. Now we can start to look at the probability of a stimulus given a bold response, right? So now we're going to look at a bold response pattern, and we don't know the stimulus. And so that is, having calculated this, we can now get at this. We can take a bold response pattern and provide an estimate of what S was. Okay? We can go the other direction. And that's why some people call this like inverting the encoding model. But this means we can decode. So that is, we can now take an out-of-set sample of time courses, and we can project it into the screen. That's what this allows us to do. Now, this all sounds very abstract. I get that. But the nice thing is what you get is something like this. So the white line shows you where the actual stimulus was on the screen. And the yellow greenish is our estimate. It's sampled from the posterior of this, where the stimulus was. Yeah, so we can actually decode per TR where our procedure thinks the stimulus was. We can, so if people talk about brain reading, in many cases they mean 55% correct for red versus green. And I want to argue that this sort of thing is a much more powerful thing to be able to do, right? Because now you're looking at visual processing in a specific region. You're really looking at it in where it's happening in visual space. Yeah. Yeah. Let me oh, I've made a mistake here. Let me go into that because you know, like I said, good questions. The answer is on the next slide. Because here we're still stuck with a model of location preference, and you know, there's much more to the world, right? And here we have an example of a study from 2008 already, where we can make more complicated encoding models. In this case, they actually added orientation. So what they did is they fit this, again, an encoding model to brain responses on natural images. And those are much more complex, right? In this case, it doesn't have color, but it does have orientation in it, of course. All these images get meaning from their orientation. I won't go into this too deeply, but this is actually done with a standard GLM approach, but then using penalized regression. If you're interested in using that sort of technique that I'm sure many of you have heard of in AI or have used in AI, this is what's being used. The point is, you can then start to decode also this. And I want to first show you an example of what does a receptive field like this look like. And then show you what you can do with this type of procedure in terms of this decoding operation that I just outlined for this simple bar stimulus. Yeah. So what does a receptive field like this look like? Well, it has a location. So this is, if this is the visual field, this is the part of the visual field that it likes. So you get that information. You can actually construct retinotopic maps from an experiment like this with natural images, and people do this. But you get extra information. You get orientation and spatial frequency. So that is, spatial frequency stands for the fineness of the patterns that are at that location. So this means finer patterns, and this means coarser patterns. So this voxel likes very fine patterns at this location with an orientation of 45 degrees. That's what it likes. And before, we had a library of all of the voxels in visual cortex location preference. Now we end up with a library of all of the voxels orientation and spatial frequency preference on top of this spatial location. Right? So there's much more information. And if you're then willing to go even further, you can add this motion direction to the mix. Right? So you can add more complicated encoding models, even explain the unit activation better and better by these complicated models. And you can then start doing something like this. If you can actually decode movies. And so this is what that looks like if you do it from the entire visual brain, right? So you present someone with these clips, and you can reconstruct from their brain something like this. Yeah. Again, the detailed operations are a bit complicated and a bit beyond this course, but you see that you can actually pick up this pattern of what the visual stimulus was like from brain responses. And I want to, this is now 11 years old, this thing. So this example. So nowadays people are doing this with neural networks. So much more intricate, much more powerful techniques that perform this this decoding operation, and that gives you you know, very lifelike reconstructions. So I want to show you that this is not just the case for sounds. Oh, sorry, for vision, but also for sound. And I'll try to, this is going to go horribly wrong again. So, you know, I'll post the But let's see. So I'm going to play some sounds and hope for the best, right? So we have sound maps in the brain. So here you see the sound map going from low frequencies to high frequencies. You can also fit these receptive field models, this encoding model. How does this any voxel analyze auditory information, and then use that to reconstruct sounds? Yeah. So here's some examples. Are they here? Oh, damn it. Yeah, they are.
Let's try again. So the first that I just showed you, or played, was an actual bird song, and the second was the reconstruction. Like, I'm not a bird watcher or anything, but you know.
Beyond all this.
But here you see an example of the actual sentence spoken, right, and the reconstruction. And you see that it doesn't do a very good job at reconstructing speech. Now, that makes sort of makes sense, because speech recognition isn't something that's very easy, right? Yeah. Yes. So maybe, yeah. If the encoding, so this is where this encoding model problem, if your encoding model doesn't capture a certain type of processing, right, then it won't be able to use this type of information in the decoding operation. Okay? So here in this case, the model that they used was just about these low-level properties of the sound, yeah? And they didn't really go into the high-level properties of the sound, like whose voice is this, or what are they saying, right? Whereas in the brain, we know that our interpretation of sounds is very much driven also by what we think the speaker is saying, for example, right? So there's a lot of very complex processing going into our understanding of speech. This model doesn't actually incorporate any of that processing, right? And so it's lost. And that's why it sounds horrible, in essence. Yeah, I agree with you that in principle, those are low-level properties, the voice and the timbre and all of that, right? So somehow this didn't happen. I would have to like go into the specific article to try to understand why they didn't capture this. My hunch is that, yeah, this is just, so it's evident that this is something that their encoding model didn't capture, and thereby also their decoding operation couldn't retrieve, right? Yeah, I'll try to look it up. So am I out of time, or do I have still 15 minutes? Yes.
Because now is when the awesomeness starts, right? Now we've only talked about visual and auditory stuff, like low-level stuff. And we've already been able to do quite a lot. I'm so glad we're inside. But there's much, much more that you can do with this paradigm. And that's really what I want to talk about now. This paradigm of trying to understand and explain what every voxel is doing in terms of its response can go much, much further, because even though we've used vision and audition as examples here, we know a lot about vision. So it makes sense that the models were created at first to explain visual responses, but people have started doing much, much more. Also, abstract dimensions, things that we wouldn't call sensory at all, have been shown to be organized in this way, such that you can actually fit these receptive fields to voxel responses, but then not based on vision or on audition, but on numerosity. So in this experiment, what they did is they had people just watch little movies of like 1 dot, and then 2 dots, and then 3 dots, and then 4 dots, and then 7 dots, and then at some point 20 dots. And then they would go back in the 7 dots, 6 dots. So very slow, like that slow bar, but now they're very slowly moving through number space or quantity space. And it turns out that there are these, that you can find voxels where the response is beautifully explained by a receptive field model that you see here on the numerosity axis. So this voxel, it has a peak response around a numerosity of 1 and then falls off to higher numerosities. This one, it has a preferred numerosity of 7, around 7, and it's very wide, right? And you also see that width in the time course. Yeah, so that's similar to, so you also have a location, the peak, and you have a size, exactly the way that the neurons have a location and a size, and the way that we have the same type of thing in visual space. Yeah. So when they found this, they didn't just look at single voxels, but the nice thing about this is that you can then say, but does this also mean that a voxel that likes the number 2 is next to a voxel that likes the number 3, right? Is the brain organized in this way, according to maps, in not the visual dimension, not the auditory dimension, but in the numerosity dimension? And indeed, they were able to find these regions here in parietal cortex, but there are more that are actually that have maps of preferred numerosity. And they could even show that a specific quality of these maps that we know of, like maps, they have a specific type of progression. Distance to preferred numerosity goes a bit like this. And we see this also happening in auditory cortex. We see this happening in all sensory cortices. Yes, so this is distance millimeters on the cortex. So as they have 1 centimeter here. So this line. Yeah, the same here, this line. Yeah, so it's low to high. So yeah, you're actually looking at this this arc on the surface of the brain. Yeah. So by appreciating the single voxel's response and trying to explain it with a simple model, what they've been able to find is a pattern, not of brain responses, but a pattern of brain organization. Right? So it really has been able to shed more light on how the brain is organized. And here's an extra quality of these maps. We know that the tuning width, so that is the size of the receptive field, but then in numerosity space, is related in this positive way to preferred numerosity. So the smaller the number, the peak of the receptive field, the more finely tuned that receptive field is also. And we knew this from vision, we knew this from audition also. It's the same for similar things hold for somatosensation. And apparently this sort of lawful relation also holds for an abstract quality, numerosity. Right? But that's the crux here. This is not just the sensation that's coming in. This is a derived abstract quantity. So this really allows us to say something about how even abstract things are encoded by the brain. Okay? And this idea of having maps then starts to reappear over and over. Because if we can go further and say, okay, numerosity, that's cool, but people are still looking at something, right? What does this mean for higher types of processing, right? It's not like our mental lives, the pinnacle of our mental achievement is counting, right? We do a lot more with our brains. So there's much higher cognition that we could also try to capture with this type of paradigm. Yeah. So we're going to try to be more abstract about it. And again, the same the same lab that did the movie decoding also did this study where they showed for 120 minutes, they showed subjects movies in the scanner. So just movies. Yeah.
A question about like all the concepts we're talking about now is under the assumption that you are showing that you are playing a sound or showing that.
Generally, yes. This is all a relation between a stimulus and a unit in the brain.
At the moment that you are decoding, you know we're showing a movie now. Or
Oh no, no.
at that point, we are trying to decode what type of sound.
Yeah. So this we only relate the bold response and the movie to one another in this training phase, right? And then at the end, like what you saw, the left movie and the right movie coming from the brain and the left movie actually being shown. But that analysis was not informed by the movie anymore. That was just read out from the brain. So that is how much of the movie can we reconstruct from the brain? That's what you were looking at. So the left movie there is just there for your reference. It was never used in the analysis. Yeah. Yeah, and we'll have another lecture on the connectivity of the brain, and I'll highlight other ways in which we can do this type of modeling where we actually don't have a stimulus, we just have the rest of the brain. But that's a bit matrix type of thing. We'll talk about that later. Yeah. Does that clarify things? Or just?
So I was thinking like the area of the brain could maybe also be active when other types of things are happening.
Yes.
And if you know that there is a stimulus, but you only don't know what type of stimulus it is, and you also have the assumption that there's this type of stimulus, but you only have to guess which type of stimulus.
Yes. Yeah. So if you have a very constrained set of stimuli, then that makes this decoding easier. Yeah. Yeah, definitely. So it's really hard, and I don't know of any examples that have really pulled it off to, for example, just let people close their eyes and think of something, and then you know, the dreaming example just goes almost there, right? But yeah, no, it's really hard. That especially if it, you know, you can think of anything. That's the power of the mind, right? You can travel to Hawaii by closing your eyes, sort of. But you get the point, and that's not what. Yeah, that's really hard to get from the brain. Yeah, so this is a like conceptual caveat that is also very important in like the mathematics behind this actual decoding, the sort of Bayesian stuff and all of that. Definitely, yeah. So just to show you what they didn't make a receptive field model of what happened at a specific visual location and what orientation and motion direction there was there. They did now is they just said, I'm just going to tally, there's now a woman, she's talking, and there's a dog, what is that, a door? I don't know. So when do these things happen in the movie? And so they would just have a design matrix, like you know from the GLM, right? Where we have 1 or 2 or 3 regressors, right? Here they have a design matrix that is 120 minutes wide and 1,705 regressors tall. But you can do a similar analysis again with this penalized regression, and then you just fit what every voxel likes. What concepts does every voxel like? You had a question? Yeah, usually that breaks your GLM. Yeah, so the general rule for standard cognitive neuroscience experiments is that you want at least 20 time points for every regressor that you add. So sort of rule of thumb, you can do statistics to be more precise. But this is like not even, you can go much further, but then you need to do penalized regression and cross-validation. If you're very interested in that type of approach, I can like maybe tack on a lecture at the end of the course. But yeah, so it's a more complicated, more machine learning based method to fit beta weights, to get beta weights, if you will. Yeah. Yeah. So I think your intuition is really valuable here. That you don't get things for free. You can't just keep adding regressors and then say everything's going to be fine. This is going to crumble down at some point. So if we do this sort of thing, we get for each voxel, we get what concepts it likes, right? Whether it likes dogs or cats or whatever. Yeah.
Is it possible that the voxel can respond on multiple concepts?
Yes.
The thing was with regions.
Yes. Yeah. Very, very, very good point. So this is not to find the concept that each voxel likes. It's much more complicated than that. Every voxel likes what is a concept, right? There you go. And this methodology is inherently complex, or at least it values this type of complexity. And I'll show you a bit about what that means. So what this means is that if you look at a single voxel's tuning, right, so you can sort of create a sort of receptive field structure over a semantic space. That's the idea. You could do that, right? And with that, you can then start to predict bold responses over time, right? So that's how you perform this type of analysis. And so what you then get is these single voxel tuning curves in the semantic space. So there is no real visual space type analogy here. You have to really think deeply about, okay, but what space do concepts live in? Any idea? No, right? And so there are methods to find this, yeah? Yes, indeed. And so what they did here, they took categories, right? And you of course have ordinate and subordinate categories, right? So you have mammal, you have dog, you have cat, right? And then you have Persian, or those really ugly, like naked cats, right? So concepts go from global concepts to much more precise concepts, right? There's a hierarchy there. And so there's one way of describing this hierarchy. It's called the word net, and there are many more. So we can talk about that as this. There's many, like in linguistics and language, like NLP type things. There are many different ways in which people decompose this type of space, if you will. So here's one. It's called word net. And you see that in the center, there's things like organism or person, right? And then a lot of things, that's a very ordinate category, right? There's a lot of subordinate types of category. And so this is more or less the structure. Like there's a bunch of disconnected things, and they're all verbs, right? Because they're not objects, right? So they don't adhere to this ordinate category that centers on objects. But so you see that there's like a rodent here, there's athlete here, there's atmospheric phenomenon there, like rain, and so like food, and then there's this structure like tools, devices, boats. You get the point, right? This is a way of taking that structure of concepts and trying to make sense of it. But it also, I'm gonna 1 minute. So what you can then do is, of course, color each of these items in this sort of network according to whether there was a positive or a negative response. And you see that you get this single voxel receptive field. This one likes things and locations. It doesn't like body parts. It doesn't like mammals. It likes persons a bit, but it doesn't like communication at all. It likes travel. Yeah. So that's how you read this. And so you get this sort of tuning curve, but it's a really complex way of looking at it. This is in the left parahippocampal place area, a region that we know responds to pictures of houses. Right? So if you do a very simple experiment and you have a house and a face, you will see that this voxel activates for houses more than for faces. And that's exactly the type of information that's already in this contrast also, right? So this gives you much more information, yeah. But it's hard to draw the line, right? Very hard to, it remains a jumble, right? You can do more, but it's also, there's a lot of complexity still in there, where it's conceptually very easy to be able to say, this is a house region, right? It's conceptually very appealing, yeah?
It could also be that this also has also some spatial
yes.
focus on objects.
No, it most likely also does. So in a separate paper, what these authors did is they fit both this type of model and a visual model to it. And they just published a beautiful paper where they show that there's like a gradient from concrete concepts to more abstract concepts, and that there's a sort of this drop off. So every voxel has a receptive field in semantic space and a visual receptive field, but they trade off. As you go up the hierarchy of the brain, they become more abstract. Yes, same thing. Yes, so more, you actually have, you have that same thing happening in auditory. Yeah. But I just wanna, because I'm out of time already, I think, and I still have too many slides again. So I wanna highlight a few things, right? Just to quickly show, like, we're still at this mess where we have this receptive fields. What does it even mean, right? So you can do some tricks, right? What you get is this brain map of how concepts are represented, right? That's a beautiful thing in general, but it's too complex, right? These receptive fields are too complex. Did I just miss your question? Oh, okay, yeah. And so it's too complex, you can't really do it, but you can do some dimensionality reduction on it, and then start to sort of color things in the brain according to what concepts they like. Yeah, so you can sort of compress all of this information. And this is then the way that you would color things. So you make things yellow if they're animals, green if they're persons, blue if they're locations, green if they're magenta if they're vehicles, and so forth, right? You also see that a boat goes together with wave, right? That sort of thing. And then you can start looking at what this looks like in the brain, right? So you can color voxels according to what they like, and then you see these semantic maps of information processing throughout the brain. And you see, for example, that areas involved in navigation, for example, I think I pointed to like magenta bluish for locations and vehicles and that sort of thing. So we know that these regions in the brain are responsible for navigation type processing, and that drops out beautifully out of such an analysis. I have way too much to say about this because it's I really like it. Oh, can we not also use science space movie encoding to find particular LTM long-term memory traces? Yeah, that's one of the. So this really relates to memory also, and that's also what people are starting to use it for. I just want to make a single sort of closing statement, and I'll try to get back to the rest of the things that I was going to talk about in a next lecture. So what I hope I've been able to show you somewhat is that with this focus on the single voxel's response, you are making your life really hard. You're really making it important that you understand exactly what's going on, that the model that you create for the responses that you're measuring is adequate, and that it covers everything that you're interested in, right? It makes everything much more complicated, much more complex. But once you have this, you know exactly what you know. You know how you understand what the brain is doing, and you understand this at the lowest possible level, that of the unit of measurement, right? And so once you have that, it also means that patterns that you measure from the brain aren't just something that you should feed into a classification algorithm and then get an answer back. It means that these patterns mean something, right? You have just created a catalog of what patterns could mean in the brain. So what this gives us also is these beautiful maps of, in general, like how the brain encodes information. And that's something that really is starting to be the state of the art. People are doing this more and more, trying to understand these patterns as instantiations of how the brain encodes information. I showed you the numerosity example. that can be a stepping stone to understanding what our brains are doing when, for example, we perform mental calculation. So all of these are stepping stones to really try to start to understand what the mind is doing in the brain, like how information is encoded in the brain in a much more sophisticated sense than we could do if we were just looking at patterns and not trying to understand where they came from. So that's the general point. And I think we're, yeah. So I'll stop the recording.